Welcome everybody. My name's Terry O'Brien. I'm the head of the School of Translational Medicine and currently acting dean of the subfaculty of Public Health and of Translational Medicine and Public Health. And welcome everyone. A warm welcome to our annual School of Translational Medicine Public Lecture. I'd like to start by acknowledging the Burong and Wurundjeri clans of the Eastern Kulin Nation, who are the traditional owners and custodians of this land, their waterways and skies, and we pay our respects to their elders, past, present and emerging, and like to acknowledge any First Nations people in our audience tonight and online on eHub. Before I introduce our speaker for tonight, I'd like to mark this as our first ever public lecture for the School of Translational Medicine. We changed our name earlier in the year from the Central Clinical School. The public lecture has been a long-standing tradition of the Central Clinical School, where we had one of our most leading and outstanding researchers talk to the public about some of the really important research that is done in the school. We were the Central Clinical School for many years, which reflects our location in sort of central in Melbourne, but it doesn't really describe what we do. So we got university approval at the beginning of the year to change the School of Translational Medicine because that really encompasses what our school's mission is. Translational medicine, for those who don't understand the term, is a bidirectional process where between clinical practice and fundamental research. And people often think of it as bench to bedside, but in fact, as important as anything is the bedside back to bench and the virtuous cycle of bench back to bedside. And much of it starts with the clinicians, such as our speaker tonight, academic clinicians, who are able to identify where there are gaps in care in our current patient care and bring it back and collaborate with basic scientists and discover new treatments and targets and approaches and then bring that back through clinical trials. And we have an ecosystem here in the School of Translational Medicine where we have the full spectrum from the clinicians through to fundamental basic research and outcomes research, and that's what translational medicine is. And it's not just research, it's also training. We have a large PhD program. We train more clinician scientists than anywhere else in the country. And the clinician scientist is one of the really key pieces of this ecosystem. And our speaker tonight is a stellar example of a clinician scientist. So I'd like to, with no further ado, introduce our speaker tonight, Professor Patrick Kwan, who is my long-standing collaborator and colleague and friend. So Patrick is currently the professor of neurology in the School of Translational Medicine. He's also the medical co-director of the Monash Institute of Medical Engineering, or MIND for short, and his clinical role is as a consultant neurologist at Alfred Health, with his main interest being epilepsy. Patrick is a native of Hong Kong and then went to the UK to complete his high school and then undertook a Bachelor of Medical Science Honours in the University of Nottingham, followed by his medical degree at the University of Cambridge, graduating way back in 1993. He then undertook his postgraduate and general medical and neurological training in the UK, at various hospitals in the UK and then back in Hong Kong. And then he was drawn back to the UK in 1998 to Glasgow, Scotland, to undertake his PhD with the famous Martin Brodie. And it wasn't just about drinking whiskey, I don't think, because his first publication from that in 2000 was in the New England Journal of Medicine and is still the most cited paper in the epilepsy field, with over 6,300 citations, and being the first line of many grants in epilepsy, including many of mine. He subsequently returned to Hong Kong as a neurologist at the Prince of Wales Hospital and clinical associate professor in the Chinese University of Hong Kong. In 2011, we were very, very lucky to be able to recruit him to Melbourne as Professor of Neurology at the University of Melbourne and head of the Epilepsy Program at Royal Melbourne Hospital. In 2017, we were even more lucky to be able to, and blessed to be able to recruit him across the river to be a professor in the newly established Department of Neuroscience at Monash University here, and also director of the Epilepsy Program at Alfred Health, which he grew to be the largest and most academically active epilepsy program in the country, before handing it on to Associate Professor Andrew Neal when he took up the role of medical co-director of mine. Patrick is recognised internationally as a research and clinical leader in the development of personalised medical treatments in epilepsy, in particular drug-resistant epilepsy. His mantra is thinking out of the box, and I think we've got a couple of boxes there to be thought out of later. And you'll certainly, I'm sure, hear that in his talk today. He's published more than 400 papers in leading international medical and neurological journals, which have been cited more than 36,000 times. He's been awarded more than $50 million in competitive grants in Australia, internationally, and multiple commercial trials as well, and commercial enterprises. He's received many international and national awards, too many to list, but including being elected as Fellow of the Australian Academy of Health and Medical Sciences, and has had multiple leadership roles within the International League Against Epilepsy. Patrick will speak for about 45 minutes, and then we'll have question and answer both from the floor in the room here and also people online. So save up your questions, and then we'll have refreshments afterwards. So I'd like to welcome Patrick to give his talk, Symphonic Science, Orchestrating the Future of Epilepsy Care.
I'd like to first start by thanking everyone. And I could have thought of no better way to start the talk by sharing with you some of the words from people who are most directly affected by epilepsy. And in their very own words, those are the words that the patient spoke to us. We did this study together with Emma and other people in the room, and Sandy. And what we wanted to hear was what it really felt like to be diagnosed with epilepsy. And as you can hear, it could be a life-changing diagnosis for many people. And I was just thinking, sitting there just a couple of days ago, I diagnosed someone with epilepsy, and it's not a diagnosis you make lightly. But in fact, it's very common. So 1 in 100 people would have welcomed epilepsy at any one time. And in fact, 4% of the population, that is 1 in 26 of us, would develop epilepsy sometime during our lifetime. So it's a very common condition, maybe more common than you have thought. And I'm saying every week I would diagnose 1 or 2 patients with epilepsy, and knowing that it could be a life-altering diagnosis for them. As I said, so sometimes it could be taking us a little more time to make that diagnosis, and you want it, and you don't always have all the information, but you want to get it right. Either way you get it wrong, there could be consequences. And it's something that for the clinicians in the room here, whether you feel the same, it could be something that's really weighing up on my heart. That's what keeps me up at night. What's going to happen to these patients that I haven't put on treatment, but they might have epilepsy? And I'll come back to that patient later on. If I can get this working, there we go. When we talk about epilepsy, most people think about seizures, but seizures really just the tip of the iceberg. And underneath, there's so much hidden under seizure, because underneath what people don't see are many things that can impact on a patient's functions. And these include impact on their cognition, and particular memory, concentration. There are issues such as mood. A third of patients have anxiety or depression. And with a seizure, there's a risk of injury, of course. And for some patients, they may even experience sudden death. And although it's rare, but it's really real. And for many patients, this is very much hidden under the surface, and that causes a lot of misunderstanding, stigma for epilepsy patients. There are many causes for epilepsy. You can see in this pie chart here. About a quarter of them, we call them sort of acquired due to an insult or brain injury, for example. But nearly more than half of the patients really don't know the actual cause, so it's not apparent. And increasingly, we know that genetic can play a big part. And seizures are also very different as well. And there are now more than 20 seizures recognized by the International League Against Epilepsy. And see here, I show some of the videos of people having seizures. And as you can see, they can range from very severe, violent kind of bodily movements to something very subtle, and just the patient becoming quiet. And it can be very hard to make a definitive diagnosis when you have such limited information. And even more problematic, sometimes challenging, is there are many what we call mimics of epileptic seizures. Many conditions can appear like seizures, and so misdiagnosis is very common. Either these conditions being diagnosed as seizures or the other way around. So the patient I mentioned earlier on, I'll change some of the details just to protect her privacy. She is in her late 20s. She's a musician. And she's starting having some events from middle of the year. Initially, very infrequent, once a month or so. But then in last month, she had 3 events in 1 week. And very description, not very clear. The diagnosis, a lot of uncertainty, not very typical. So we thought, oh, let's bring her to the hospital, do some monitoring. Oops. Like this one. She came in, have all the wires on her head, and she had to put up, very inconvenient. Just imagine this being the gold standard these days. Patients to come in, they have to arrange work and workplace, maybe family and friends, look after their children. And worse still, the beds can be cancelled in some hospitals. won't name which one. And it's very frustrating. Worse still, I think, is the patient have to put up with hospital food, and doctors like me pestering them every day. And I don't know which is worse. And so this is not a very accessible method. But she did come in. She stayed for 4 days. We didn't catch anything. No event. EG was entirely normal. So what do? We let her go home. But then 2 days later, she had another episode. By this time, it became clear that she definitely has developed epilepsy. So we put her on a medication right away. We thought, okay, here's a script, go and take it. And she was taken by a very caring husband. On the way home, while in the car, she had another seizure. And I couldn't help but just imagine, and hopefully she wasn't driving, but what if she was driving? If she had an event and an episode? And the consequences could be just beyond I can imagine. So this is not working. This is not for every patient. So what is the current gold standard in the community? And unfortunately, we are stuck in the Stone Age most of the time. And this is how we monitor seizures in the community in the form of a diary. Either it is a paper-based diary or electronic. But regardless, whether it's paper or electronic, it still relies on the patient being able to record their seizures or the family and friends having witnessed the seizures. And for a lot of patients, they don't have someone with them 24/7 a day. So you can imagine this is not a very accurate method. And indeed, in this study that was performed in Melbourne here, led by Professor Mark Cook at St. Vincent's Hospital, Professor Terence O'Brien in the audience here, and Professor Sam Berkovic at the Austin, when they recruited 15 patients, some I would say very brave patients, to have electrodes implanted on their brain surface, as you can see on the left-hand side. And these electrodes record EEG directly from the brain surface. So nothing can escape. This is the truest of the truth, if you like. And look at the slide on the right-hand side. You can see on that graph, what it shows is that there's a huge discrepancy between what the patient themselves report and what actually recorded on these electrodes on the brain EEG. And for most of the time, there's most of the patients underestimated the number of seizures they had. And so some patients, you can see, they thought they had no seizure for that whole month, but in fact, they had 200 seizures. They were all missed by the patient. And you can imagine how difficult it then becomes to either make a diagnosis or monitor whether the medication or treatment is working for the patient. How do clinicians know whether to increase the medication, or is it safe to decrease the medication? Or think about whether the patient can return to driving if they have no memory of their seizures at all. And how do we, and this is, of course, groundbreaking in itself, demonstrating this significant problem. But again, it's still an invasive procedure. You need an open brain surgery to have these electrodes put in the brain. And so we are very excited that Professor Mark Cook has went on to develop a newer method and started together with a startup, so that the implantation of the electrodes they don't need to actually go inside the brain, but rather is using a small device. And I've got in my magician's hat here, just like this. So the device can put inside just underneath the skin and connect it to another device outside that picks up the signal. And the signal is then transmitted through the cloud, through an app, and made available to the treating neurologists and the patients as well to decide whether any medication changes are needed. And we're very proud to have recruited a number of patients in this clinical trial. But what this study is showing here with Dr. Hugh Simpson leading is to assess the effectiveness of such a system. Does it really help improve patient outcome? Does it reduce seizures? Does it improve the patient quality of life? Would it make it easier to reduce medication toxicity on the patients? And this is a groundbreaking study in itself as well. And we're very much looking forward to see the results. Nonetheless, this is still a level of invasiveness. It's less invasive, but you still need to undergo a surgery. It's not open brain, but it does require a little bit of a cut in the scalp. So can we do better? What about wearables? Can we put things you just wear all these wearables and Fitbit and so on? And we know there are these products around. The problem with them is they are very uncomfortable. I don't have a Fitbit or Apple Watch, and one of the reasons is it's just you have something on your wrist all the time, and it's not that a lot of them have not been validated for detect seizures. So what we've been working is with Professor Wen-Long Chen and his group, who are chemical engineers, and who used to be based in Monash now and moved up to University, is to test a material, a new form of material called gold skin. And essentially, it's a highly stretchable materials. And you can see here there.
The material is ultra stretchy and comfortable. The final product will be fitted with Bluetooth and a battery to deliver immediate results to a smartphone.
And so you can imagine we can have one of these, again, out of my magician's hat. This is the device, just like a piece of plaster, Band-Aid. The Band-Aid can put over your elbow or put on the patient's wrist, and they can wear it anytime. Once it's used, you can just peel it away. And this provides a much more comfortable way to monitor seizures. And we are currently analyzing or evaluating this new sensor in our video imaging monitoring units, like you saw earlier on, having patients will wear this and EEG as well to correlate the changes and to validate the data we collected. And we've got AI scientists like Dawn sitting in the audience here going to look at the data for us. Now, you might say to me, this is still, I don't like wearing things. It's too much hassle. I can just stick things on, charge stuff. Can we do even better? Can we do it with any kind of things that the patient needs to wear? And this is a project led by Dr. Shobi Sivabambu in the audience, and together with Dr. Deval Mehta, who is an AI scientist at Monash University, to analyze videos to monitor seizures. And when you think about seizure video analysis, you might immediately starting to get worried about what about privacy. We're all worried about concern about privacy when we when all these problems of facial recognition. So what they've done is to convert the raw video images into these other features, such as body pose or facial landmarks, or looking at optical flow, so that they can be analyzed in a privacy-preserving way. And we're currently, again, trialing this, developing the algorithm, But the way it might be applied, you can imagine, is once it's developed, then it can be a monitor or camera can be off the shelf, may be placed in the patient's bedroom to monitor if they were having seizures during sleep, which is a very common concern for patients who sleep in their own bedroom. What if I have a seizure while I'm sleeping? And that's almost a very often weighing on the patient's mind. Or you have someone having an episode, your family and friends, and then they can take out the phone, take a video recording, and have that analyzed automatically. Well, once we've made the diagnosis, the next step is, of course, to start treatment. Now, when I was a medical student, and maybe even when Terry was a medical student, he had even fewer choices. He was down there in a hospital. And life was easy as a neurologist. There weren't many choices. Once you try the field, you just stuck with the old one. But look what's happened. Terry, in the last 30 years, we've been working as doctors. Look at this, there's been an explosion of new anti-seizure medications developed to treat our patients. And that's fantastic. That's all the research that's gone in, billions of dollars to develop these new medications. And they've given us far more choices, more options to choose to tailor or individualize the best medications for that particular patient. But it also brings with itself a different problem, which is that how do we choose with now so many different medications to choose from? And to illustrate this, I need volunteers. This is the part of audience participation. So I've got here a number of balls, all right? Who wants to catch the ball? But Ben's got the tallest arm, so catch. Oops! Oh. You like that ball? Does that work for you? It totally works. Excellent. Got it right the first time. Who wants to catch the ball? Don't be. You like sport. All right. We'll try tennis. Well, how's that for you? Okay, let me try something else. What should I try? I've got a few here. What about a little footy, a red one? Take this, I hope I'm not upsetting any of the farmer friends here. Take levetiracetam. You like that?
It kind of helped a little, but not quite.
Not quite. All right. Well, we need combination therapy. How about second drug? It's a squishy one. This is a new mechanism of action. You've never seen this before, never tried it. Whoops! Oh, sorry. Missed completely. That's what happens. Does it work now? Oh, I'm so glad. Great. Thank you. You can keep it. So you see, sometimes it can take 2 or 3 times to find the right job, or even more attempts. And worse still, some patients, if one were to try the field, they still won't come under control. And that's what happened to one of my other patients. I always were still being seeing us and still trying to control his epilepsy. He is in his 30s, was holding a very respectable professional job as a financier, have a good income, and very happily married. But then he started to experience seizures about 3 or 4 years ago, and we tried different medications. I think we tried 3 or 4 different medications. We tried other things except the kitchen sink. Eventually, we needed to undergo epilepsy surgery. And I would, unfortunately, I still have to say it's still work in progress to bring his epilepsy under control. And so there are some patients we need to look at non-pharmacological, other forms of treatment other than medications. But we don't know when they would require it or whether they need to use this other form of treatment. So what happens is, as you can see in this analysis by Dr. Ben Chen, who's in the audience somewhere, where he spent 6 freezing months in Glasgow, eating haggis and fried Mars bars occasionally to see through all the data of nearly 1,800 patient records. of these patients who were newly diagnosed with epilepsy and followed up for some even up to 30 years to look at what happened to these patients long term in terms of their seizure control. And what they found was eye-opening, of course, because at the end of the follow-up, 36% of patients, that's over a third of patients, were still not seizure-free. They were still having seizures despite trials of different medications. Another message from this pie chart is that for most patients who did become seizure-free, they did so on the first or the second, maybe a little bit on the third treatment, but after failing 2 or 3 treatments, very few patients became seizure-free. But then and these patients are those that we would recommend to undergo sophisticated evaluation for other forms of treatment, such as surgery or neuromodulation or dietary therapy, which we all offer at Alfred here. The problem is they have to endure seizures by trialing medications that are ineffective, sometimes for years before they get offered these forms of therapy. So wouldn't it be nice if at the very beginning, I would know that Shobi is going to need the combination therapy, or my patient would need a surgery at the very outset, rather than having them trial ineffective medications with the risk of side effects. And again, this is actually not a unique problem for epilepsy. And in any conditions, you can almost guarantee for a group of people who seem to have the same condition, given the same treatment, you have 4 different groups of outcomes, depending on whether the treatment is effective, is working, is useful, or whether they're having side effects from the treatment. And this very unfortunate patient of mine developed a very severe form of side effects in the form of severe skin reaction and high fever, which can be life-threatening. And the happiest patients, of course, are those in the top left corner. When the treatment is effective, there are no side effects. And the most miserable patients, I guess, are those on the bottom right, when they have the treatment is ineffective and they're having a lot of side effects. So how can we find out earlier on how the patient will respond to the treatment? And this is again, I guess, the goal of what is called precision medicine, which is trying to understand the genetic differences between individuals as a way to predict whether some treatment would be effective or they will develop side effects to other treatments. And this is the goal of precision medicine, which has been a lot of work in the last 20 years or so. And in epilepsy, we do have some examples. And for example, these very severe reactions, now we know there is a genetic variant that can predict people who would develop such a severe reaction. So we can do the test first before prescribing the medication to avoid these very terrible side effects. Now, this all sounds very well. We now can do the test, can predict. What's wrong with this? Well, the problem is that implementing the test, this precision medicine approach, has a lot of barriers, both logistical and financial barriers. And that's because genetic testing has to be done in the laboratory with very specialized equipment by highly trained staff. And you need to take a blood and then send it to the laboratory. So imagine last time you had a blood test, when you saw your doctor, what happened? Well, he decided you're going to do a test. He wrote the pathology slip. He gave it to you. You then went to the pathology lab. And some days or weeks later, you go back to the doctor to get the result. And this might be fine for conditions or test results that are not so urgent. But for conditions like epilepsy, where every day, if not every minute, might count, you want to start treatment as soon as the diagnosis is made. This causes major problems in the management flow. So what we need is a test that can be performed at the point of care or on-site that can deliver results immediately within the clinic consultation. And I'm now moving on to my second white box. And I'm glad to say that after years of research, we've finally created such a device, a system that you can run a test using a small card. I poked it. That's it. Something just a small card like this. put your sample in, and then pop it in the device, touch the screen a few times, and then the result displayed within 30 minutes. And in this way, the right prescription can be prescribed to the patient before they leave the clinic. So rather than waiting for a few weeks, they go for a cup of coffee, and they come back with the results and the script. This is, we're hoping this is going to solve a lot of the testing problems. But it won't solve everything, because epilepsy, as I've said, is such a complex condition. There's so many factors we consider in choosing medication for the patient. And increasingly, we are turning to artificial intelligence, or AI. Let's take a breath. AI is scary. We all heard a lot, it has had a lot of bad press. But AI can allow us to look for connections and relationships that may not be apparent. In particular, deep learning has provided us the tool to uncover hidden connections. And it's almost, epilepsy, I think, is almost a perfect example to apply AI, in particular as a support for the clinicians to make clinical decisions. And we've recently developed a deep learning model to predict whether the treatment will be successful for a given patient when they're prescribed with the first anti-seizure medication. And to do this, we included clinical factors. And these include the age, the medical history, which is very important in deciding treatment selection, and also results from the tests such as EEG and MRI, and put this into the model to come up with a prediction of whether a treatment would be successful or not for that particular individual. And we utilized data from 5 different centers from 4 countries around the world. You might say, that's all very well. How do you know it works? The only way to show is to do what we should do for any medication, any intervention must be prospectively demonstrated, but prospectively tested rather than just relying on retrospective data. So we're now testing this model in a trial that we call the Personal trial across Australia, involving more than 13 different sites. And so adults who develop epilepsy would be separated into 2 groups by randomization. One group would receive the usual care, where the neurologist would choose the medication for them, whereas the other group, the neurologist will be provided with the machine's recommendation, and then they can use that information in their prescription. And importantly, we'll follow up the patient's response, not just about whether they're having seizures or side effects, but even looking at whether this is going to be cost effective for the healthcare system. Is it going to increase costs or reduce costs? And I think even more importantly is to look at whether such a method would be acceptable to the patients and the neurologists, actually. And what would be the ethical impact of prescribing medicine in this way? What would be the people's concern be? And this might be about trust. Can you trust what the model recommends? What about what about does it interfere with the relationship, the doctor-patient relationship in this sort of application? It's almost like having a third person sitting in a consultation room. And we've also heard some other feedback. The neurologists are worried that they'll be de-skilled. They may not know how to prescribe drugs anymore. If they, just like we may not know how to drive anymore, if we sit on Tesla for too many times. Moving on to something different in terms of personalization. Who can tell me what these are?
Ben cells.
Ben cells, yeah. Ben's and Jingchao. So what you're looking at are photographs of neurons or brain cells that are being cultured in a laboratory. In this direction, maybe just 1 floor up, literally. They are growing in the incubators. And these cells, these brain cell neurons, they are, I think, the prettiest. They're like the stars in the sky. You can see the strands and connecting, just like the nerve brain cells in our brain. But what's interesting about these nerve cells or neurons is that they are actually derived from stem cells. And how do we get these stem cells? from the patient's blood sample. So in our laboratory, led by Ben and Anna, we've established a way that you can just collect a blood sample, and using the blood cells can then be turned backwards, if you like, turn the clock backwards to become stem cells. And then the stem cells can then be differentiated into different types of cells to study. It can be heart cells. In this case, we're interested in the brain, so we derive them into brain cells. And with these brain cells, we can do a lot of things. And the most important is because they come from the individual, they retain all the genetic information of that individual. So it becomes a model of the living brain in that particular individual. And these can be used to model the condition, the neurological condition the patient may have, or even to use this to screen compounds or drugs that might be effective for that particular individual before we apply it to that person in a much safer way, because then the patient wouldn't have to take the risk of, for example, side effects, taking medications that would not work for them. And as I said earlier, because the hallmark of epilepsy is electrical changes. So what we do here is it's like doing EEG on the nerve cells, if you like. So we measure the electrical activities of these cells using electrodes. And in this setup, we have 12 electrodes in a well, and that well can contain up to 100,000 neurons. So each electrode would measure activities from the new electrode surrounding it. And you can see here that every now and then there's a burst of activities. You can see on the blue here. And these the activities recorded from each electrode, when they burst together. So meaning that a network has been formed between these nerve cells, just like our brain. And this is recording from a healthy individual. So what do you think it would look like from a patient? And this is an example here of the recording signals that we recorded from a patient with a genetic form of epilepsy. And you can see here that the bursts are more frequent, they're closer together, they're a bit shorter, but they fire much more rapidly, showing that there is increased what we call excitability of the brain cells, which is, again, as I said, the hallmark of someone having epilepsy. Therefore, they're prone to having seizures. Now, to have a better appreciation of what it really, this increased activity is like in our patient, what Ben and I did was to pass on the signals, this file, to Dr. Matthew Walsh. I'm not sure whether he's listening online, but Dr. Walsh, or Matt, we call him, is a computer scientist, a software engineer, a neuroscientist, and also happens to be a musician. And that combination in my dictionary is a genius. So he's a genius, and we said, what can you do with it? And one of his many skills is to turn signals into sound in a process called sonification. So sonify the sounds or even into music. So would you like to hear what the music sounds like? So what do you think the music from the patient sounds like? Now this one, you would hold on to your chair. What words came to your mind?
I felt hyperactive and anxious.
Anxious.
To me, it sounded like being underwater as a person with epilepsy, like not being able to, like, it just sounded like to me, I can't get out of this. I need to breathe. I need to breathe. That's what.
Like they're trapped.
Trapped, like needing air.
Yes, fighting.
Yeah.
It's chaotic. A lot of energy. And whenever I listen to it, I still get goosebumps. Because I actually listen to in the car on my way to work. This is why I can't remember in the morning. But I can only imagine if this is what's going on in my patient's brain. No wonder they are having such a big impact on their cognition, on the psychological, mental health. No wonder. Well, this is just to show you we're not just doing all this to make impactful music, and in fact, we're using the system, as I said, to screen for new drugs. And this is one of the projects that we are collaborating with Dr. Laura May at the Monash Institute of Pharmaceutical Science, or MIPS, together with Ben, to try to discover new class of drug that utilizes our brain's natural defense system against seizure, if you like, by targeting a protein in the brain called adenosine. So adenosine is a protein in the brain that rises during a seizure to stop the seizure itself. So it's our brain's natural antidote for the seizure. And what Lawrence Lab has done is to create a library of compounds that can enhance or increase the effect of adenosine when the seizure happens. And the beauty of this is that it only works when there is seizure. So it avoids, at least in theory, some of the side effects. And we are currently using our stem cells, derived neurons, to screen for these compounds and pick the winning ones to then go on to do animal studies before clinical trials. I hope at this point I've convinced you that we really have moved on in terms of the research technologies, the approach from the industry 1.0 to the so-called 4.0 age, where we are much more using interconnected technologies, going to stem cells and AI. And I haven't even talked to you about many of the very groundbreaking work being done on this very campus by other research leads, such as Professor Terence O'Brien's research and other people's in the precinct here, in trying to develop better treatments for our patients. And this we have really propelled, I think, in terms of technologies available to us to make new discoveries. But now I'm speaking to the researchers, the scientists in listening, the audience, and I sometimes wonder whether the way we do research, we are still stuck at the 1.0, the factory line approach. You can call it multidisciplinary, yes, everybody is doing their thing, but in fact, it's not. They're all working in silo. There's no support between the team members. Everyone does their own thing. The group leader is never going to go to the lab, is he? So in fact, this is a very dehumanized, impersonal. So what we need to do is really work together, more like an orchestra, where we support each other, we play together to create really the symphony, and that would have a sound nice to the audience. And if you imagine, some of us, we all come from different skill sets, we have different expertise, of course, but it doesn't mean we can't play together for this harmonic symphony. And if you imagine, we have the violinist sitting over there, the flutist is outside, the conductor is absent, the music would be awful and no one would listen. So we must play together, come together, and in this process, the consumers are paramount. It's essential that we involve patients and their carers in this process. They must have a table, or a seat on a table, sorry. And I'm very proud to be co-leading with Dr. Neha Cole, who is a dietitian and early career researcher in our group, to start what we call the Epilepsy Consumer Advisory Panel. So advisory panel that's dedicated to support epilepsy research, both basic, translational, as well as clinical research. And we'll be launching the event for this next month. Mark your diary. You can scan the QR code, and I hope to see many of you at that event as well. What we need, I think, to do also is further out of the box, is to collaborate or to work together and start thinking about not just about who are your peers or your consumers, but we must think of how can we move from innovation to commercialization. And sometimes as scientists, the C word, commercialization words, sounds a bit dirty. We are scientists, we are kind of not like this, an ivory tower. But if you walk into the hospital, when you look around, it's been said to me once that apart from the people there, everything has been commercialized. If things are not commercialized, they cannot be used, they cannot benefit patients. And at Monash Institute of Medical Engineering, we have developed a suite of training and support for researchers to advance their ideas from ideation to design the studies to providing some seed funding and further on to go on to commercializing their research project to link them into potential investors and the capital to make what they do into reality, to really make an impact on people care. For those of you who don't have epilepsy, you don't know anyone with epilepsy, you're not doing research in epilepsy, you might say, why should I care about epilepsy? And this is work done by Dr. Emma Foster, sitting in the audience here, taking a picture. She's a neurologist, research in our group. And she showed us that epilepsy, why we should care, is because epilepsy has huge impact on how productive a society is. And individually, epilepsy has greater impact on an individual compared to some common conditions, such as diabetes and hypertension. And with more than 100,000 people at working age living with epilepsy in Australia, epilepsy costs us $32 billion in GDP loss, productivity loss from these patients over their lifetime. The good news is, if we can just make 5% more of patients seizure-free on Ben's pie chart, if we can move that down just 5%, we don't need 10, 5% more, it would save hundreds of lives. It would lead to thousands of years of life gain, and it would save hundreds of millions of dollars of medical costs and save the society nearly $4 billion in GDP lost over the lifetime of this individual. To finish off, as my favorite neurologist, Arthur Sachs, said once, it is the genetic and the neural fate that defines our uniqueness. And so we must move to the age where we, rather than treating everyone with one-size-fits-all or try and error, to move to a personalized management. And I hope what I've shown you is that this is now in sight, I think, for epilepsy for the first time. But we need to do more. We must collaborate more broadly. We must engage beyond our comfort zone. And this will bring benefits to not just people with epilepsy or their loved ones, but the entire society. And I want to acknowledge all the people. I'm so proud to be working with a team of very dedicated scientists who try to improve the lives of people every day in this campus. And all the collaborators, of course, all the people with epilepsy, family carers who have participated, and of course, those people entrusted us with their funding to do the research as well. But we need to do more. How can we do more? And what we're envisaging is a new platform that will bring all our efforts together to make really major breakthroughs that our patients deserve. And we imagine this new platform will help us unify the research efforts across multiple schools and campuses of Monash, so that we can go to really foster innovation, advanced education research, so that we can achieve that ultimate goal that every person living with epilepsy can live their life to their fullest potential. So what do you think? Step away from your production line and join our orchestra. I can't promise you that we'll solve all the problems, we'll discover answers to everything, but I can guarantee your neurons, my brain cells will be singing together. Thank you very much for listening.
As I absolutely anticipated, that was completely out of the box. and out of the hat as well, and absolutely amazing, visionary, creative, fun. You know, that's really congratulations, Patrick, on an amazing lecture, an amazing, absolutely stellar career. We're very proud you're one of ours. So we've got a bit of time for questions. We can take questions both from the floor and also online. Does anyone want to start the bidding? You have one over here? Yes. We've got roving microphones for people.
Professor Kwan, my name is Cassandra Scott, and I first of all wanted to thank you and everyone else who's here today and online for your lifetime commitment to this work. I'm here today as a guest of Phil, but I have a very personal reason for being here, and that's because I have genetic epilepsy, and I had all kinds of different medications prescribed, and they were mostly ineffectual, and that culminated in me drowning and being found in the ocean with no heartbeat for more than 15 minutes at Coogee Beach in Sydney. I was successfully resuscitated, as I'm standing here today, and then I changed medication and became seizure-free, and I've been seizure-free now for more than 11 years. So and nowadays I have had the great good fortune of following lived experience, and my day job is now as I am the Assistant Commissioner of the NDIS Quality and Safeguards Commission, and so I'm looking after the quality and safety of people with epilepsy and all other types of disability. So I just wanted to say that my entire life has been changed by having got the correct diagnosis of pharma to me. So thank you very, very much for making that change to as many other humans on the planet.
Now, like questions, or while you're thinking, Graham Shears, who's the CEO of the Epilepsy Foundation, said, Patrick, there's two questions actually. Firstly, does the gold skin detect only convulsive seizures?
That's a good question. So the first instance, we're developing the appropriate analysis for convulsive seizures. But you can imagine it can also test for non-convulsive seizures. So for those, for less kind of technical jargon, convulsive means there is a shaking kind of movements, if you like. Non-convulsive means usually the patient is for quiet, sometimes another sort of blank seizures or absent seizures. And the reason this may still be possible is because we know during, even without the bodily movements, there could be changes in autonomic function or skin conductance that can be picked up by these kind of sensors. So these can be incorporated in the sensors to detect non-convulsive seizures as well. So the potential is there.
Right. And Graham's second question was, there's an impressive number of new drug treatments, but as you point out, the rate of seizure freedom has not materially improved since the 1840s when I went to medical school. Is there, what is your hypothesis for why this is so?
Well, I have two hypotheses. One is that I still believe, I still don't believe we should give up on all the effort we've done in many, many years. And so one hypothesis is maybe we're not using them in the correct way. And maybe, as I said, we're just not picking the right medication for that individual. And we know some patients just give up once they try 2 or 3, they just don't want to try anymore. And so by using methods such as AI or by using a stem cell neural platform, that will allow us to personalize treatment for that patient. And so it will allow us to a different paradigm to pick the right drug. The second hypothesis, of course, is that none of them have really targeted working in the right way. And this way, to solve this problem, we need to find medications that may have new mechanisms. And I think by using a stem cell platform, it can allow us to do high throughput screening, complete out of the box, really, drugs, compounds that we may not have thought of with any effect. But this provides a platform that can allow us to screen them in a high throughput way and a safe way as well. And I would also add that even so the proportion of patients seizure-free doesn't seem to have changed, this is a very high bar. In many patients with the new medications, they get seizure reduction, and which can be very significant for many patients that are having fewer seizures, to allow them to do things they would otherwise not be able to do, or medications that have fewer side effects that are less problematic. For example, for women who want to have children with drugs that now have lower chance to cause birth defects in babies when the mother takes some of these older anti-seizure medications. So the intention, obviously, then was a very kind of stark message, but I would say that in reality, the new drugs they have come along and have many advantages and have really, I think, allowed us to choose medication in different ways. So the benefit is not just all about making no seizures, but it has other benefits as well.
Questions in the room? Yeah. I was going to be very disappointed if you didn't ask a question here. There's never a seminar that asks a question. And one over there, okay?
And thanks to Patrick Kwan. It's a very enjoyable talk. My question is about the curious about the progress of the epilepsy and treatment itself. So my understanding, for example, some other cancer or the other things like you treated, it can be a recurrent, it just come back again, or something something at the beginning is very mild and then become more severe. So I just, out of curiosity, just want to know how is the nature of this development progress of epilepsy and the treatment. Do they come back? Like sometimes you don't know, like say, okay, we are seizure free for 10 years and then suddenly just come back, become more severe. Is there such a scenario? Thanks.
Yeah, that's a great question. And in fact, indeed, that can be the case for some patients. So the cause of the condition can fluctuate. And some people can go through a period of very good seizure control and then have more seizures as a cluster. And a lot of work have shown that some seizures actually come in cycles. Maybe what we're seeing are actually cycles of seizures. And I think the challenge compared to other fields, such as you mentioned, oncology, is that the brain is not as accessible as a piece of tumor that can be resected from the patient. And so this creates a lot of challenge, how we understand what's going on inside the brain. And the only way, safe way to obtain tissues from the brain is actually when we do epilepsy surgery. And that's invasive in itself and not many patients eligible. So I think really think that models like the stem cell neural models can provide us with that, at least a model, somewhere to start, a building block to build hypothesis and understand what's happening in the brain. I would say also that there's a lot of effort looking at multimodality, multi-omics, and we are learning a lot from the oncology field, for the oncologists listening. We are very behind in neurology, at least in epilepsy, I can say, but we're trying to catch up.
Good. Now, there was another question. Yeah.
Yeah, Patrick, Justice Mangaro, CEO of NeuroGPT. Thank you for letting me ask a question. There's touching on just the other question that was asked before me. My sort of roundabout question is that there's a number of different approaches to tackling the problem, and I can see the value in hitting it from various ends. You're advocated trying to make drugs more effective, which is a terrific approach. There's also what do you do with the other patients where drugs are never going to work? And then the other question is, well, do we really understand what's going on in the brain in the first place? And there's probably a relation between all of those. And so the question I wanted to ask was, can you sort of comment more broadly on the various spectrum of approaches that can be used to try and treat epilepsy and whether we're focusing on all of it, how do we focus on all of these things? And on top of that, what can we do systematically in the way that we triage and decide on treatments for patients, which can take years and years and years before people finally get recommended for implant surgery, which can also be a big impact.
Yeah, I mean, that's a broad, very great question, Justin. I think what we in clinical practice, what we do is we take in the whole person, and that's we still say making a diagnosis. Writing diagnosis is still a very much clinical diagnosis. We rely a lot on history, witness account, what happens in a seizure. We put in all strands of information, test results to try to understand without getting into the brain, invasively understand what's going inside the brain, and that's really, really challenging. In a way, if you flip it the other way around, you can say despite all this lack of knowledge, we're operating like in a black box. We managed to get 70% of patients seizure-free. That's pretty remarkable. So they're looking at the other way, as a half glass full, we can say, in fact, we're doing better than someone would expect. But obviously, for the 30-something% of patients not seizure-free, then it's a struggle. And I think the approach is, so far, what we're taking here is different multiple prongs. I think the approaches to improve diagnosis, how can we diagnose and monitor the seizures as well, it's been a long-term effort and using different technologies. In terms of treatment, other than medication, as you mentioned, there are multiple other forms of treatment as well. And in a kind of a healthcare service paradigm is how do we increase awareness? How do we make the treatments available to be available to the broader patient and even the doctors and the GPs and so on, because many patients are still managed by the general practitioners and general neurologists, and they're not even often referred for assessment. So we have in the international community a definition of drug resistant, meaning failing of 2 medications. That should trigger referral to special centers like ours here in Alfred to evaluate what kind of best treatment. And even that, I think there's a lot of work in terms of education and increasing awareness among both the patients, their carers, as well as the medical professionals.
Great. Final question, Chris French. Welcome to this side of the river.
Great to be here. Strangely enough, I have to give a talk tomorrow on reimagining epilepsy, so I came tonight especially.
It's all online.
So it's hard to ask a good question in this context with all your experience, but just one thing that phenomenologically, just get back to every now, I'm sure you think about it a lot, is as opposed to most other medical conditions like heart failure or cancer or things like that, there's a really disorder, there's an absolute disorder in the organ system. But in epilepsy, the brain works just fine almost all the time, with the exception of the more severe epilepsies. And that's such a striking phenomenon that we use drugs all the time and they work well, in 70% of cases. But have you got any comments on that approach that most of the time it's normal? Is there some way that we can just treat it when it's abnormal, or can we detect that?
That's a good point, Chris. I would say what epidemiology tells us is that, in fact, the brain is normal most of the time. And that's why you increase comorbidities. That's why if you look at some literature and work done by Emma here showing even people with third seizure, they have high rate of depression, anxiety, cognitive impairment, and also people have. That's why there's an increased risk of developing all these other neuropsychiatric conditions. So I think what that tells us, my interpretation is, in fact, the seizures is just, as I showed in that slide, it's just tip of the iceberg. And maybe we shouldn't be chasing after the tip of the iceberg all the time. We should look at what's underneath. And how do these take a more system biology approach and looking at these are brain conditions, but they manifest differently. Are there any final common pathways? Are there shared pathways between different conditions? And the way we think about the brain conditions, a lot of these is we box them based on what we observe for thousands of years. Epilepsy has been described in millennia, and we're still using that very phenomenological approach to treat and manage and understand it. And that's a very artificial way. Perhaps I think we need to look at, that's why I go beyond just tackling the seizures themselves.
So as our final question, we have Lisa Todd from Epilepsy Action, who's online and going to be unmuted, I understand. Lisa, we should be able to hear you, I think. Ask your question.
Patrick, thank you so much for such a wonderful talk, and it was very entertaining, even though it was online. I was wondering if you've got any indications from consumers living with epilepsy or people living with epilepsy, their level of acceptance of AI prescribing, and also how long it takes for the neuron, the cells to be growing for the personalised medication testing.
Thanks, Lisa, for that great question. I wish nobody could be here because I would have thrown the ball to you. But you can't do remotely. No other way to sit anyway. I think that's a really good point. And so in the AI work, we are collaborating with philosophers at Monash University. I never in my life did I think I would collaborate with philosophers in research, but here we are trying to understand what's the ethical impact of AI, and we are trying to understand it from all the stakeholders' perspective, from neurologists as well as from patients, consumers, as you mentioned, and some of what I've heard from the philosophers, the consumers' view are very eye-opening again. You know, on one hand, we sometimes as researchers or clinicians, we may assume that all patients, they will be very scared, very against. But in fact, some patients take a very pragmatic view and say, you know, if this car is going to keep me safe on the road, why should I trust Patrick behind the wheels? They are quite open-minded in a somewhat surprising way. But what's often a consistent feedback is that they would like to involve their neurologists. So when neurologists in the audience, we'll probably keep our job. That's good, because they want to have that endorsement, acknowledgement. And I think, again, what that means is that I think in this room, maybe 5 years ago, we thought, oh, AI will never affect my working lifetime. And I think there's no hard to deny that AI is already making an impact. And it's often said, it's not that AI will replace the neurologist or the doctor, but the AI will replace, but the doctors who use the AI will replace those who don't. So I think we need to embrace and look at how to utilize AI to help patients and work out all these ethical concerns. With regard to the neurons, I'm looking in my the most talented scientists in the group here, looking at Ben, working in the lab, how can we make things faster? And I have the same question. I always want things to happen yesterday. So how do we speed up the process? And I'm sure with time to come, we'll work out a better way to develop the model perhaps. We don't have to wait until they're fully grown. So I think at the moment, probably it'll be hard to implement for someone with newly diagnosed epilepsy, but for someone with chronic epilepsy, Johnson epilepsy, when we have a little bit more time, then it will may allow us to implement this system. And we're doing research in collaboration with University of Queensland to do just that. But we can screen drugs using the stem cell neurons first, and then trial them on the patients.
All right, well, we could chat with Patrick forever, and we're going to have refreshments outside, so just join with me again and thank you, Patrick, for the most amazing public lecture. And I also want to thank Liz, Janet, Nash, and Mary and Kirsten for the fabulous work they've done behind the scenes organising tonight's lecture. These events are always so much more work than you ever think they would be, and you guys have done a fabulous job, and Loretta, of course, as well. Thank you in the organising, and please join us for some light refreshments.